So I guess my first question, Stan, is how did this all happen?
Well, I'm not going to go back to the very beginning. But anyway, so most of my friends or colleagues don't realize that I had an early interest in agriculture. And I won't go into why, but I worked on citrus branches in high school, and then when I actually got my, after I graduated from UCLA, which was after World War II, I went to work on a big farm up in the San Joaquin Valley. And then when I thought of going into graduate school, my interest really was in agriculture and actually most likely in plant breeding. And so when I applied to Berkeley for graduate school, which I didn't know whether I was going to get in or not, I told them what I was interested in, plant genetics, plant breeding, and they said, okay. And so I was admitted. And so I started graduate school in 1949, and went through all sorts of things, got interested in everything, in evolution, population genetics, so my mathematics was too weak for that. And probably one of the most inspiring experiences at Berkeley was a course from Kurt Stern in human genetics. He was quite an impressive lecturer, and he was very interested in the subject at the time, and I remember that quite well. But then when I finished in '52, well, when I found out that there weren't that many opportunities around, it may even happen to some graduate students these days, but there was, and I just sort of cast my net far and wide. I wrote to a number of people in all areas, and since I was interested in human genetics, I thought, well, I'll write to someone in that area too. And one of the people I wrote to was a man named Franz Kallmann, a German psychiatrist who was interested in schizophrenia. He did quite a bit of twin studies. And actually, he was one of the few people who gave me a positive answer and suggested I apply for a NIH postdoctoral fellowship. I don't know quite what they were called in those days. And I got it. And I'd never really thought much about that until Charles was asking me something about these early days. And I said, how in the world did they give me, with this background sort of in agricultural interests, with no real human biology background except, maybe a course in zoology and a course in human genetics from Kurt Stern, did they give me a fellowship? Well, I'm glad whoever was on it decided to give me one, because I don't know quite where would I've been if I hadn't been. But if I'd been in that committee, I think I would have questioned whether I was given Stan Gartler's fellowship. At any rate, Franz Kallmann was at the Medical School of Columbia in New York City. And so Mary and I were quite excited about going to New York and staying there for a while. And so we got there in July of 1952. And I won't go into any great detail, but I remember the first day I went into the lab, and there was another postdoc starting at the same time, a man named Gordon Allen, who was well trained in medicine and psychiatry and was really quite appropriate for his position. But I went home that night after that first day talking with Colin and other people and told my wife, I may have the shortest academic career in history because I don't think I'm going to succeed with this person. At any rate, I wasn't. We didn't get on very well. And fortunately for me, Theodosius Dobzhansky and L.C. Dunn, who were at the downtown part of Columbia University, they at the same time were interested in setting up an institute for the study of human variation. Dobzhansky especially had the idea there was going to be loads of polymorphisms to be found, and Dunn was also already working it out. And so I went down to see Dunn, who was a terrific person and quite a diplomat. And I'll tell you one question he asked me. When I came in, he said, "Have you come here to complain about Coleman?" I said, "No, I just want to escape." And so at any rate, actually Coleman was a very bright guy, but we just did it. But anyway, so Dunn arranged everything that I would become part of the institute, and Coleman would still remain my supervisor in a way. And so I ended up working for Dobzhansky, trying to look for polymorphisms, looking at urinary amino acids and twin studies. So that was and I'll tell you just one story about that. So the first paper I published with Dobzhansky, it's probably trivial but interesting. He wanted to make sure that there weren't significant dietary effects of these excretionary patterns. So he put himself in a diet of apples only for I don't know how long. And he'd bring in the urine samples out of one. And so while he was doing that, I was thinking, well, I'm going to look at something else. I'm going to eat all sorts of odd kinds of food and see if anything changes. And so I ate a variety of things, and one thing I ate was dates, and I found a new ninhydrin spot appear, and it was quite clear. And so he got interested about this, and we did a little work on it, and soon I realized there was a paper we were publishing in Nature, just a short paper on this rather trivial, even I considered it trivial. And I realized then that the name of the authors were probably pretty important, whether you got something published in Nature or not. At any rate, the paper was published saying, this new thing. And within a few days, the newspapers in New York, one of them picked it up and said, professors at Columbia have discovered how to determine when a co-ed has a date last night. And that was, anyway, we had a great 5 years at Columbia because everybody came through that institute who was working in the field of human genetics. So we got to meet lots of people, and it was a great thing. And then the man who saved me is sitting there, Arno. He came through Columbia wanting to bring some people out to Seattle to work in this new division of medical genetics, and so he picked me. So I ended up out here. That's how I got here.
Stan, you stated this decision about going into human genetics so matter-of-factly. It's a crucial question that graduate students still come, of course, to us and ask. What were you thinking at that time? I mean, did you ever imagine going into plant genetics and deciding that human genetics was more interesting? Or?
No, in fact, I obviously was quite naive because, as I said, I still can't imagine why they gave me a fellowship. But at any rate, and I don't know. Once I got in, people didn't question it, and I read like mad to try and get caught up on human biology and. Well, even working with Dobzhansky on a urinary excretionary amino acid patterns, I did a lot of reading because I hadn't had a good background there. So I was naive, but somehow it didn't seem to bother me too much. Maybe it was having been a GI in World War II. I don't know, something like that. But I was foolishly relaxed about it.
Well, obviously it wasn't a foolish outcome. So tell us how the department emerged from this convergence of Arno and Herschel.
Well, let me show you. I know we're not having any, can't show slides, but I'll pass this picture on, which is my favorite one of the department. You won't be able to.
Let's get on the film here. So that
you can get on.
Yeah, is that okay? Can you see that?
Up a little bit.
Okay, thanks.
So when I came out to join Arno in 1957, Herschel had already been here a number of years. He actually became a member of the department, I guess, in 41. I think before the war, and I went in. And I should mention, by the way, as far as an early history of the department goes, the article by Dave Stadler is great, and I'm sure many of you have seen it. And so when we came out, when I came out with Arno, so we had our own small group in the Department of Medicine, and present in Seattle at the time were Herschel, of course, Dave Stadler, and Don Hawthorne, who had been a student, and Howard Douglas in microbiology. And so this was 57, and then there were a number of things that went on when Herschel hoped first to try and alter the whole structure of biology at the university. And he was thinking first that we should have, instead of a department of botany and zoology, which were the only departments at the time, to have a division or a department of biology with division of genetics and so forth. And there was an attempt to do that, but it didn't get through the zoology primarily maybe botany. They in fact were against it. And then things went on, and Herschel said he was unhappy and stuff, and so they finally, the university finally gave him a department of genetics. And so that was in 1959, I guess, formally, there was a department of genetics. And within a couple of years, there was a committee set up, and I think the first person brought out was John Gallup, if I'm not mistaken. I think that's right.
Major mistake on their part.
Now, I remember Herschel made a special trip to Johns Hopkins and spoke with your advisor, whose name I don't remember now, and went after him. And then Larry Sandler came out to teach a course once again, and he was brought in. And Ben Hall, I remember, came out to be interviewed by physical chemistry, or the chem department. And I remember seeing a famous paper of his in Science, which Herschel hadn't seen, and I alerted Herschel to it, so Herschel got to work on Ben, and so Ben joined our department. And so the department grew quite rapidly for a while, maybe not even fast enough for Herschel's taste. And he was then, I believe, offered the chairmanship of Caltech. And so he said, well, we need something more than the space they'd given them up at the upper campus in what was formerly botany. And so together with Neurath, the university said you could apply funds for a building. And so we got the building that sits over there, and more appointments that was set up. And so the picture I'm passing around now, maybe I just grab it back for a second.
Let me get
Oh, yeah, questions. Okay, thanks. So Herschel, this picture is of the department in 1979 when Herschel had retired. At that time, when you turned 65, you could no longer be chairman. And so the department and the regular members are all here except for two. There's Gus Dorman, Dave Stadler, Carol Sibley, Arno, Herschel, myself, Ted Jones, Don Hawthorne, Ben Hall. You can see through a number of people. Unfortunately, it's a little sad that Gus and Dave and Herschel and Don and Larry are gone. But this was the department at that time, and it was quite striking.
Stan, those early years of genetics, at least from my perspective, having come in '66, were very exciting, and the department had some unique properties from the experiences I had had, the cohesion and could you tell us what features and how they developed that were particularly effective in the graduate education?
Well, I think one of the main things when we started, even before there was these other members, we set up a journal club, which Herschel at first was actually kind of opposed to, or not, I would say he wasn't fond of, but then he became quite enthused with it and insisted that it become a very important part of the thing. I remember when we began, we didn't have enough people in the department to form, and I remember people from biological structure, biochemistry would join with us, and we had a very good group, and then as we grew, I think the department mainly probably fostered and encouraged and led by Herschel's ideas, is the department tended to set up sort of rules as to who could be a member and who couldn't. We had a number of people who wanted joint appointments, and Herschel was quite willing to consider them, but he wanted the joint appointees to act pretty much like they were members of the department. They should attend journal club, they should attend seminars, etc., and things like that. And I remember there was a lot of discussion about this, and there also were people who didn't want to go along with that. But I think his ideas were important in the early days of the department in keeping it together, very packed. Like he didn't want a student to go too far from the department for their PhD. Mm-hmm. And in fact, when at this picture, when Herschel stepped down and they were looking for a new department, I remember there was a committee was set up to talk to various people about how to make the new choice. And I remember, I don't know how my colleagues were interviewed, But when I was interviewed by the committee, they made a big point of the fact that under Herschel, the department had been too inward-looking. And I felt, I didn't use those words, but in my opinion, Herschel was right. It was important to build up this department, to keep it together, to keep the graduate students so they knew where they were going and what their aims were. And I think it was a very important point. And I think the inward-looking thing that the committee had very much in their mind, I always think was a little bit of envy, because the department had already become rather well known. People got certain honors. And of course, today the department is just, under Bob, is so totally different. It seems to cover everything in a way. And I realize that the department as it existed under Herschel for 20 plus years, it probably couldn't exist anymore that way. But I think for the students that went through, whether they were in my lab or Larry's lab or Lee Hartwell's lab, I think the basic pattern and the ideas that Herschel set up were quite important for those students. And that's sort of my views in present.
The quality of those presentations was extremely high in my recollection. How did the department keep that quality so high? Were these students coached? Who was responsible?
Oh yeah, you were asked to, if you had I remember I had one student who was extremely nervous, and I don't know if he ever forgave me for this, but I insisted he take a course in public speaking over campus, and he actually improved a great deal. And I said, "Gosh, you're doing so okay." But he said, "No, 1/4 is all I'll do." but then we had of course a fantastic student, Bruce Baker, who I'm sure many of you know about. And Bruce must have been born with qualities of being a professor, really great professor, because the first journal club he ever gave was professional. He knew everything, and his the way he presented was terrific. And you knew if some student was having trouble, if Bruce took him under his wing, his next performance was improved. He really was quite extremely impressive. Yeah. And Larry Sandler, of course, was always there in the audience, always making comments. And if somebody put up too many graphs of a particular technique, which Larry didn't like, the student heard about that. You had to have, and of course there was chalk and stuff like that. So the journal club, and there was John Gallen with his special. By the way, I forgot one important thing now that I saw, John. There's 2 people missing from this picture because everybody was still alive at this time. And there's Howard Douglas, who was a wonderful man, and I know he was around because he gave a great talk that evening at the dinner, a wonderful talk. And the other person missing is John Gallen. But John Gallen tells me he was here, but he says he doesn't photograph well. So I don't know where you were, John. Anyway, so our journal, and I thought one of my accomplishments, I was always trying to get, although we have plenty of participants, I was always trying to get postdocs to participate. And it was difficult at times. And I finally succeeded in getting Maynard Olson to give a talk. And it was, I think, about a month before he was leaving for wherever he was going. And he came and said to me, "Stan, I really apologize. I should have been doing this much earlier." And so the Journal Club was a great thing. And then there were all sorts of other things we had. It was quite good. Yeah.
Stan, tell us about your own research while you, starting from your time here. What were pivotal events and papers and techniques that really caught your attention, and how did it?
Well, when I came here from Columbia, it was '57, and I was doing a few things still with excretion of urinary amino acids and ideas of going into somatic cell genetics were developing. And that's one of the, and Arno had advised me, he thought cell culture was a coming thing, so I went into cell culture and set various techniques up. But I'd say the sort of major thing that influenced me and led to my doing some worthwhile things in the early years was 2 things. Was the exon activation explanation of dosage compensation in humans, and the fact that females were mosaics, and also the marker glucose 6-phosphate dehydrogenase, which Arnold was interested in and Ernie Beutler had done some wonderful work on. And so what I sort of realized at the time that I wasn't particularly interested in dosage compensation or that aspect, but that the use of these markers very early in development, and you have somebody who was a heterozygote, that it created a mosaic which gave you cell markers which you could use for tracing out various aspects of development, fate mapping, which I guess the word wasn't quite used at that time. And that really became, as I look at most of the things I've done, especially in the first 10, 15 years, that they were all aspects of that. In fact, a postdoc who came to work with me, Dave Linder, who most of you won't know, but he was a practicing pathologist in San Francisco. And he had become sort of tired of just the heavy work of hospital pathology, and he had obtained a fellowship to take off for a year and just do research. And he came up to the pathology department here, and he was a very independent type person. And Earl Bendit, who was head of pathology, told him what he was doing and said, just go along and talk to people to see what they're doing, what you're interested in. And although I wasn't in pathology, one day he turned up at the lab and introduced himself and asked me what I was doing. And so I told him all about G6PD mosaicism and how we could use these cells as tracers and stuff. And so he essentially took off his jacket and said, let's get to work. And so he immediately introduced me to tumors that I didn't know anything about. And so we did some other things first, but then we quickly got hold of tumors. As a pathologist, he could go and get any kind of material he wanted to. It wasn't like today that you had to file this or file that. He just walked into the room where they were cutting up people and took people that were dead. And took whatever he wanted then. So we quickly looked at this tumor, this leiomyoma of the uterus, and showed that we could show with the G6PD markers that they started from single cells. And so that led out to a lot of things. And then Phil Fialco, who was a young, I guess, instructor or assistant professor at the time in the Division of Medicine, he got quite excited about that. And Dave said I encouraged Dave, he could have really gotten almost any sort of position he wanted, but they went back to San Francisco in hospital pathology, which he finally said was a mistake, but he did that. And so Phil Fialco took it over, and Phil really ran with it like a 100-meter Olympic sprinter, and he eventually became dean of the medical school. So he did a wonderful thing. And then we've had a number of some of the grad students worked in different aspects of that. But I would say that was my major thing, among other things.
You mentioned Phil Fialco and Linder. I was always amazed to see the variety of fellows and students who were coming by your lab, some to work and some just to visit. How did that balance come about? That is, the graduate students and the medical fellows.
Well, I think that's one thing that Herschel set up, that the graduate students' careers, they all circulated through the labs in their first year. And so they actually got to know the faculty, and I think in general they felt quite relaxed about going to visit and mix. So I think that was a very strong feature. Mm-hmm. And I find, if I look back over the grad students, postdocs, they've been extremely impressive in many ways. In general, I tried to, when they joined the lab for thesis, I tried to figure out problems that they could call pretty much their own. Mm-hmm. Probably one of the most interesting grad students that also indicates a lot about the whole department was a fellow named Gary Thorgard, who came around in the 1970s, 75 or so. And he'd gone to the University of Oregon, he had quite good grades, and he came up to take a PhD in fisheries. And he was unhappy with fisheries, he was there just a short time. And he came to see Herschel, could he switch to the Department of Genetics? And Herschel looked over his record, and I think there was a meeting saying he was fine. And then Gary said, but he said, I want to work on fish. And so, and Herschel pointed out to him, said, well, there's nobody here. We have people working on a number of things, but we don't have anybody who works on fish. And Gary was a very, very determined person, and so Herschel said he would think about it. And so he thought about it. He said, well, there's Stan Gartler, who was really in agriculture, and switched to humans. What's the big deal about going to fish? So anyway, so Gary joined my lab, and he convinced me. And so he actually discovered the first sex chromosomes in trout or some kind of fish or something. He did very well. He actually became chairman of biology at Washington State. He's still there, producing lots of papers. He's a very, very effective person. And I realized that the lab got on quite well. Mike Liskay, I think Mike was still in the lab at that time, and everybody was happy about the thing. So we had a lot of great students, and they put in lots of ideas and things of that sort.
How about your teaching, Stan? What are some of the highlights in terms of graduate, undergraduate teaching, the colleagues that you taught with?
Well, I think in teaching at the graduate level, the main thing we had this wonderful course with George Martin and Don Pius on somatic cell genetics. And I think that personal people, like Dave Cox was one of the ones who got most out of that course. And Dave went on, he was working with Brian McCarthy at the time, but then Brian left, and so he finished his PhD with me. But Dave went on to do all sorts of things, very, very active things in the Bay Area. But Dave was a terrific student in that course and kept it going in many, many ways from the student standpoint. So that was, at the graduate level, that was my main teaching.
Mm-hmm.
At the undergraduate level, I taught a course for non-majors in human genetics.
Mm-hmm.
And that was interesting. It was quite, I still remember one special, the class was rather large at one time, and I could tell there was a fair amount of copying going on. And I remember one final exam, I was sitting there, and it was clear that a male student was looking at a female student's paper. And so when she got done early, she came down and she said, "Did you see what was happening? What are you going to do about it?" And I said, "Well, I know what's happening, and I'll look at the papers." So I got down, she essentially had flunked, and he was obviously a very bright guy and must have been laughing just to her. So I was glad that I was chicken and I didn't go and pick him up by the scruff of the neck. But undergraduate teaching was great, and I really had a relatively easy time with it. It was just fine. So we had lots of things going on.
I've always been impressed with your research collaborations, the facility with which you bring those to fruition. What would you tell grad students and postdocs? How did you make it work so well?
I don't really know. It just, if you're interested and you're both interested in the same subject, then it was just talking with Phil Fialco and it's interesting, Arno, who I've known now for ages, I think we've only published 2 papers together in probably 50 or 60 years. But the way I feel I've collaborated with him all the time. And but with other people like Dave Linder and the postdocs, Liz Kaichis, who was a very, very, very impressive postdoc. Does an awful lot of things, an awful lot of good things. I found it was just a matter of talking with people as much as you can and try to interacting with it as much as you can. And I would say, if you go back to the one thing that I did, I guess completely on my own, was the stuff with the HeLa cell, which is attracted a lot of attention. That was an early day. It was back in the 60s, and people were trying to push ahead with somatic cell genetics. So I had a particular idea that I was going to look for somatic crossing overs, which I wanted to do. And so I picked up a lot of lines and looked for them for proper heterozygous markers. And so I just started collecting as many cell lines as I could, and people send you whatever you want. And then I found out when I started checking them for markers, they were all the same. And so that came out to be that one story. And I never got to the stage of detecting somatic crossing over. I don't know if that would have got as much attention as the helocell contamination story did.
Yeah.
So you told me a wonderful story about Herschel's response
Oh, yeah.
when you told him that, he asked you what you were going to talk
Oh,
about.
yeah. So Herschel and I were great friends. We were great family friends. And he knew I was going off to this meeting where I was going to talk about the helocell contamination. He didn't know what it was about. And he said, where are you? What are you going to this meeting? Where is it? And what are you going to talk about? And so I told him, and he looked at me and he said, really, really serious, he said, "You're going to talk about cell contamination at a scientific meeting?" And so I said, "I guess I am, yeah." Nobody will even care about it or listen to it. And so I quickly spent a couple days putting together a lot of other things, which of course nobody was interested in. So Herschel, of course, didn't realize what the state of cell culture was at that time. No, that was odd.
How do you see the field of genetics as it's changed, Stan? What are some of the highlights of these changes, and did you recognize them at the time?
Well, the sequencing is just beyond me. I still remember, when I was doing undergraduate teaching, and I would ask my molecular friends about, are we going to be able to do DNA sequencing? And there were many people over the years who said, well, no, the pyrimidines appearing they're too much alike, we're not going to be able to sequencing. So, that's what I would tell classes I taught at the time. And of course, the sequencing and the new generations are just mind-boggling. It's really it's sort of beyond me. And of course, looking at so many other things, it's very hard to imagine. I find myself much too limited to being able to handle it. So what I might mention just a word, in order just to keep my scientific sort of sanity about me, I spent a little time with Marshall Horwitz's lab, who was down at South Lake Union. And Marshall actually his major interest, though not his major work, is in fate mapping. And whereas that, as I mentioned, that was my earliest interest, but we had very limited markers, G6PD, maybe one other. Marshall thought of the idea of using somatic mutations as trackers, and that means then you aren't limited at all if you have enough of them. And so, and that I find I can handle, I can sort of conceive of and think about. And so I play with that now. I call it play down at South Lake Union, and it's nice because you can sort of deal with the whole thing. On the other hand, when you look at the maze of markers you can deal with, complexity of them, and I realize that as young people, if they can handle it quite well, that's what impresses me the most as it goes on and on.
This is, I want to ask one great question that Stan Fields had suggested. This leads up to it, and you get it as an elder statesman in genetics and biology. Where do you see biology heading?
Towards explaining everything. No, I it's very difficult as you get older to not become sort of skeptical about what new techniques are doing. I tend to become more and more impressed with them, but there's always a certain degree of skepticism as what we'll be able to understand. But I would think that besides sequencing, as we understand proteins and all that, it's just, I think everything will get more and more, will explain more. Whether I will be able to understand it, that's a big question. Mm-hmm. One of the things I want to mention, I've got, this was the 50th anniversary of the X activation idea. And so I went to a meeting and gave a very brief talk about the history of dosage compensation. And in preparing for that, I read some of Muller's papers, which I hadn't read before. And in 1932, he gave a big talk on mutation, but as part of it, he talked about his ideas of dosage compensation. And when you read him talking about how he looks at the gene. This is in 1932. It's really impressive because you can see this man's mind thinking and almost knowing what we know today, which he had no idea about. Because he was thinking of just how dosage compensation comes about, how the X chromosome in the male, how its transcription, he didn't use that word, is enhanced, and just how and I realized that if I had met him at that time, I would never have gone into the field of genetics because I realized I couldn't possibly understand it. But he really had a insight in things. And I suspect a man like Muller today could give you a very good answer to what you're asking. I'm not sure I can, but I think it's going to be great.
Well, you talk about massive data collection, but you've also talked about the comprehensiveness of the sets. And what role do you see genome sciences playing in this transition?
Well, it's sort of the I'm going to say the opposite. That's not the word. It's I guess in a way a natural expansion of what Herschel's ideas were about the department, which was rather confined, and of course even Herschel, bright as he was, probably couldn't see how technology was going to advance so rapidly. But I would say that it's grown so and has so many appointments of individuals who can contribute to all aspects of the field that I feel is going to be the source of some awfully important findings and discoveries. Very, very strong. And how to keep, I would say would be difficult is keeping a broad department like that, which is so broad, keeping it focused. Like, I look at medicine, which was first a member, with Arno has been since 54 or so. There, I think medicine suffers from the fact that it's so broad. It's so many subunits, divisions. And I would worry whether that could affect the field of the Department of Genome Sciences. And then you can get some fracturing. So I think the struggle between keeping it together, which Herschel was always focused on, and sort of the natural centrifugal force of having so many areas tending to separate it is something that really has to be looked at and difficult to handle. But I think Bob can handle it. Or at least he'll try, that's for sure.
Stan wondered yesterday if there would be some questions, and I've tried to save some time for questions from you folks.
I have a question. Going back to the journal club, journal clubs are very common in many departments. I've sat in on journal clubs in other departments, and I am always amazed by how extraordinary the presentations are in this department. I wonder what you think about the history of that. How did it come about? There's something in the culture of the department that has had a big impact on the way the journal clubs are done.
I think I mentioned earlier.
I want to repeat this so it gets on the tape. So John Gallant was asking about the quality of the journal clubs and how the history may have helped foster this excellence in presentations.
Well, as I mentioned, we put a lot of emphasis on it, and when students had difficulty, I told you that Paul Kratzer, I sent him to take a public speaking course, but and then other students would help the other students, and I think faculty tended to give them previews of their talks, and so there was a lot of emphasis on that, and Larry Sandler was very gentle, but he always let them know what they were doing wrong, and he would have them sit down with him, not necessarily his own student, any student. They would often come and he would sit down with them and talk to them about the proper way to give a good talk. So I think I think the pattern was set, and I know with my own students, I would always check with them early on. What are you going to talk about? What paper are you picking? And I would discuss whether I thought it was a good or bad paper, and we would have previews at times. That's what I recall, and I think it was done rather well. And you always gave great talks, and the trick with your chalk, that was always anyway.
Very clear.
Yeah. So there are 2 points of similarity, or at least 2, in your history and mine. One is that your first exposure to human genetics was from Kurt Stern. And the second was that you came to Seattle because of Arne Matulski. And I'm wondering if...
I thought you came because of me.
I came to be used. and probably lost my voice, if you remember what you said. I wonder if the ease with which you began to do human genetics is attributable in part to the fact that both of those scientists, both Stern and Matulski, saw human genetics as a branch of genetics, and to them it was a natural way of thinking about human biology, rather than being something separate from the rest of genetics.
Well, I think that's true, but I would go back earlier.
So that's a great point. So Mary Claire is making the point that Stern and Dobzhansky saw genetic...
No, Matulski. Dobzhansky too, but I was
and
thinking about
Dobzhansky
right.
too, yeah.
And Dobzhansky too.
Saw human genetics as a branch of genetics
Right.
instead of medicine.
No, and I think I was going to say that even earlier at the Institute for the Study of Human Variation, which really was great, because not only was there L.C. Dunn, who most people have never heard of, but he was the one who was author of the book, Synod Dunn and then Synod Dunn in Dobzhansky.
He came in and out of Berkeley a lot.
Yeah, he was a terrific guy. And then they brought people, there was a wonderful man named Cipollini, an early immunologist, who was there for a year. And Ruth Sanger was there for a period. So I got to meet loads of people, and they didn't ask me about my background in tomato genetics. And so it was a fantastic opportunity to learn and to get involved. And we had and all sorts of people who came through. You know, Dobzhansky and Dunn knew everybody.
Right.
For example, we had just all sorts of people who come through who were interested in genetics. The anthropologist Ashley Montague would come through, and then the people working in blood groups, Alexander Weiner, Philip Levine, all sorts of people. So it was a great opportunity.
So.
Yeah, I wondered, what do you see as the big differences between, or similarities, between the way that back then, in say the early 50s, at the time that you got to that you came here, in the differences of the way that you thought that genes, chromosomes, and cells operated compared to today? Did you back then have a fully modern view of the cell? And if not, in what way?
So Joe's asking if you had a fully modern view of the cell, and if so, in what way, and what was it?
Well, I certainly didn't have a fully modern view of the cell. You know, there are so many things that have appeared over the years that we didn't know about at the time. And I think in a way, I personally was probably hindered by the fact, you know, that the field of human genetics became captivated when we finally could count chromosomes and look at chromosomes. Mm-hmm. And I think, though, it was very important from a clinical standpoint. I think from a theoretical standpoint, I think it held us back, because it really, chromosomes were nothing to the people in mouse genetics and plant genetics. And so in human genetics, we spent an awful lot of time finding this abnormality or that abnormality and writing it up. And I got caught up in it to a certain degree. So I think in a way that held us back in the field of human genetics to a certain degree, although it was important. But I think once that passed, then it seems to me today, and even way before that, that workers in human genetics are essentially aren't any different from people working in Drosophila or any other organism. So I think now that there are no real differences in my opinion.
Following up on Joe's question, just thinking about it, do you remember when you first learned about DNA as a double helix and its consequences?
I was, well, when I got my degree in '52, the faculty at Berkeley still argued about whether DNA was a hereditary material. I think that probably most of them felt that way, but there was no. And then when I got to Columbia, then of course the paper came out and we all knew about it. And so it was, whenever it was published, we talked about it. But, it was an interesting point you bring up. Dobzhansky was a very good friend of Alfred Mirsky's. I don't know if you remember that name or not. But Mirsky was a famous professor at Rockefeller. And he was one of the strongest opponents of the idea that DNA was the hereditary material. And it took him several years before he finally said he was wrong. And so Dobzhansky never, while I was there, he didn't accept DNA as the hereditary material because Mirsky had said, proteins are more important in many ways. And but as far as going to seminars and talking with grad students, it was widely accepted as far as I remember. It was. There wasn't much you can.
So what was your favorite set of experiments that you did that you can remember?
Stan's asking, what's your favorite set of experiments that you've done?
Well, I would say I guess the markers showing that the leiomyoma came from a single cell was the cleanest and the simplest type of thing. But probably one of the more interesting ones was back before that, one of the main experimental observations that led to the X inactivation idea was the fact that X autosomal translocations, if you had an X autosomal translocation and you had a marker in the autosome which didn't variegate when it wasn't translocated, it did variegate when it was translocated. And that was some of the major observations that both Mary Line and Lyon and Russell had. And so I thought it would be cute to look for an X autosomal translocation in humans. This was in very early days. I don't think anyone had found one yet. And so I said, well, I'm going to look for this by looking for an individual who has two different eye colors, assuming, and the evidence was good that eye color was not X-linked, but it was autosomal. So I said, I'll find somebody with two different eye colors and then look at them cytologically and we'll have an X autosomal translocation. And so I mentioned, and I was quite uninhibited at that time. Everybody I met, I said, do you know anybody with two different eye colors? And a young pediatrician said, well, there's a little baby on the ward with two different eye colors. I said, well, great. So I told him what we're doing, and he quickly got blood from the and it turned out that this baby's blood, there was no translocation, but she had half XY cells and half XX cells. And so she was a chimera, in fact, the first human chimera, whole body chimera that was found. And that was, I liked that experiment. And looking back at it now, I'm embarrassed. There was the pediatrician who had made the observation. The girl was a fellow named Sam Waxman. His boss, his name escapes me right now, was the head of pediatric endocrinology. And then Elo Giblett worked on the blood groups, and there was somebody else involved. So within about a year and a half, there were 5 papers published on the same subject. As I look back, a little embarrassing, but it was a cute experiment. It was fun.
I have a question. So, Stan, of all the things that we now take as sort of commonplace in the field of genetics, what was the greatest prize for you at the time this discovery was made, whether it was yours or somebody else's?
Well, I think...
So Bonnie was asking, what was the greatest surprise in genetics?
Well, I don't know if it's a surprise, but certainly, because I for years, I'd accept felt DNA was it. But when the Watson Crick story came out, and the simplicity and beauty of it, I would say it'd be pretty hard to surpass that. And there were all sorts of things that kept coming up, but that was quite amazing.
Stan, I have one question about Dobzhansky. The evolutionary synthesis was, I guess, almost over, but that is, he wrote his book in 1937, and Fisher and Wright and Haldane had been writing in the 30s. By the time you got to Dobzhansky's lab, was evolution still a hot topic that you were playing with in your discussions? And how did it play out in terms of your approach?
Well, not so much things we were doing. We were doing such simply practical things of making sure the paper chromatography worked, and we didn't. And our neighbor, we were in a 1 or 2 floors of a building, and the fumes from our paper chromatography were always rising up to some department that complained about it. But so we were filled with simple things. But Dobzhansky's students, it was quite a mixture. Dick Lewington was a student in Dobzhansky's lab at the time, and he took part in everything that was going on. And Tim Prout, I don't know of anything about Tim Prout. But there were, Dobzhansky had a great group of people. And then there were other, the department at that time, Herb Taylor was still there, Francis Ryan was still in the Department of Zoology, and there were numbers. So I would say that, we didn't really work ourselves in the evolutionary area, but we were always part of it at the seminars and things. And Dobzhansky was still was quite involved. I've been on the question of X activation, which of course is the dosage compensation form in mammals. I've been doing a little just historical work, and I was amazed to find that Dobzhansky has a paper in 1957 under Sophilus salivary chromosomes, in which he shows that the male X is twice the diameter of a single female X, or equal to 2 paired female X's. And he proposes that this means that the male X is working twice as hard to produce and bring about dosage compensation, which a view which Muller later on, although that was his original idea, he disagreed with. But anyway, so Dobzhansky must have been doing that work in his fly lab while I was around, but I never heard anything about it.
Yes.
Stan, what first attracted you to X inactivation as a topic for studying?
Well, it was the main thing was the use of cells which you could use as markers for trackers of development. That was the whole thing. And so we looked at a number of things early in development, and we looked at just how were tissues formed. We would look to see what is the size of a patch or a clone. And so it was a matter of getting our techniques for electrophoresis down, cutting out teeny bits of tissue, extracting it, and we did all kinds. For example, to pick something simple, we looked at hair follicles. See, could we determine whether the hair follicle began from one cell, from a huge number of cells, and by looking at the variegation we'd find in the heterozygote, we actually were able to determine that it started from a very small number of cells. And we did a number of things with that. So we looked at the composition of blood cells, of different kinds of white cells. And so we did a number of papers of that sort. And that was the major thing.
Stan, I fully expect that in 10 years, that it is on the 20th anniversary of the Genome Science Department, I fully expect we'll be back to ask you more questions about your current research.
You'll be back for sure.